Why should the fluidity of cell membranes be important to cell function? We've already spoken of one reason, namely the ability of cell membranes to function properly depends upon the ability of embedded proteins to move laterally within the fluid lipid bilayer. There's another reason though, and this has to do with the stability of the membrane itself. We'll begin with a simple calculation. Let's imagine that we have a simple lipid bilayer separating the extracellular fluid from the cell interior. This membrane is very thin, on the order of 5 nanometers or so. There's another thing about cell membranes, and that is that they maintain a separation between the very different internal and external environments. Within the cell, for example, the concentrations and compositions of the various ions is quite different from what it is on the outside. Potassium concentration inside the cell, for example, is considerably greater than its concentration outside the cell. The reverse applies to sodium ions. Internally, the concentration of sodium ions is very low, whereas the concentration of sodium ions outside the cell is higher. The upshot of these concentration differences is the presence of a relatively steady voltage difference that's typically maintained across the cell membrane. It's quite small, on the order of 50 to 100 millivolts or so. To make our calculations easy, let's just say that the voltage difference across the cell membrane is 100 millivolts. Now, this is a voltage difference, but there's another way to express this as a so-called voltage gradient. The voltage gradient is the ratio of the voltage difference, delta V, and the distance over which the voltage difference exists, in this case, the thickness of the cell membrane, T. So let's do a quick calculation of what the voltage gradient is across the cell membrane. It is 100 times 10 to the minus third volts divided by 5 times 10 to the minus ninth meters. When we resolve this ratio, it comes out to be about 2 times 10 to the 7th volts per meter, or about 20 million volts per meter. To give some meaning to this number, let's make a comparison between the voltage gradient across a cell membrane against the voltage gradient you typically get in a thundercloud. Now, opinion differs on this latter figure, and estimates vary from about 100 volts per centimeter up to about 1,000 volts per centimeter. In other words, the voltage gradient within a thundercloud is on the order of 10,000 to 100,000 volts per meter. These are voltage gradients that are 2 to 3 orders of magnitude smaller than the voltage gradient maintained across a typical cell membrane. This is more than just a clever calculation. It has deep implications for the stability and function of cell membranes. We've already pointed out that membrane fluidity is important for the function of cell membranes. Specifically, membrane fluidity is important for allowing embedded proteins in the fluid lipid bilayer to drift about laterally within the membrane. Membranes simply cannot function if these proteins are locked into place by excessive viscosity within the membrane. But now, what about those massive voltage gradients? We remember that the volt is really a measure of potential energy. That is, it's an electromotive force that acts to drive flows of charge, like ions. As such, we can redefine the volt in energetic terms, namely as a joule per coulomb. What the voltage gradient across the membrane tells us is that there are 20 million joules per coulomb of charge per meter trying to drive ions and other charged materials across the membrane. The membrane stops this flow, and the charge can only flow by prying apart the lipid bilayer of the membrane. Thus, the voltage gradient represents a very strong threat to the integrity of a cell membrane. The membrane meets this threat by high cohesion forces acting between the phospholipid molecules. These high cohesive forces are what makes up the membrane's viscosity and that impede the membrane's fluidity. Thus, the membrane has to strike a balance between having sufficient fluidity to allow embedded proteins to move about laterally within the membrane, but sufficient cohesion to hold the membrane together against the large voltage gradient across it. A membrane that's too fluid, that is, that allows embedded proteins to float around easily within the membrane, is also a membrane that is susceptible to disruption by the voltage gradient across it. On the other hand, a membrane that is built with sufficiently strong cohesion to completely resist the disruptive force of the membrane voltage gradient may be a membrane that is so viscous that it locks its embedded proteins into place. To function properly, then, the membrane has to strike just the right balance between fluidity and cohesion. Returning now to temperature, the problem is that temperature affects this balance between cohesion and fluidity. Here's a graph of membrane viscosity versus cell temperature from various kinds of animals, ranging from Antarctic fish to mammals. As you can see, increased temperature reduces membrane viscosity. That is, it reduces the cohesive forces between phospholipid molecules in the lipid bilayer. Thus, an increase in temperature might make the membrane more fluid, but it also may make the membrane insufficiently cohesive. Increasing temperature, therefore, can make the membrane more susceptible to disruption. A decrease of temperature, on the other hand, increases the cohesive forces between membranes, but this may make the membrane too viscous for its embedded proteins to move about with sufficient freedom. So, we can pose a hypothesis, namely that the composition of membranes should vary with temperature so that the membrane's fluidity and viscosity are properly balanced over all temperatures. There's a formal name for this idea. It's called homeoviscous adaptation. We can explain homeoviscous adaptation in this way. Suppose we have a plot of membrane fluidity versus temperature, like the one that we introduced for melting point earlier. Let's say that we have a series of phospholipids with ever-increasing lengths of the hydrocarbon chains. Each of these phospholipids has its own melting point, with each characterized by increasing fluidity with respect to temperature. Obviously, the longer the hydrocarbon chain length, the higher will be the melting point, and the lower will be the fluidity at any given temperature. Let's now lay out our range of ideal membrane fluidities. Membrane fluidity that is greater than this will be too fluid, and membrane fluidity that is lower than this will be too viscous. It's in this ideal zone in between that membrane fluidity is properly balanced between cohesion versus fluidity. We would predict, therefore, that as temperature increases, one should see membrane composition shift to include phospholipids with longer and longer hydrocarbon chains. Of course, length of the hydrocarbon chain is not the only thing that influences membrane fluidity. The degree of saturation, that is, the extent to which the hydrocarbon chain of the phospholipid lacks double bonds, also will affect fluidity and melting point. Specifically, phospholipids with saturated hydrocarbon chains should have a higher melting point and lower fluidity compared to phospholipids with unsaturated hydrocarbon chains. Again, if cells regulate their membrane fluidities to stay within an ideal zone, as we get to higher and higher temperatures, membranes should come to be composed of phospholipids that are more and more saturated, that is, that have fewer and fewer double bonds within them. Okay, we've made our prediction. Now, does it have any validity? The answer to that question is both yes and no. This question has been studied extensively in fishes, so let's take a look at some data from them. This study compares brain cell membrane phospholipids from two different fish adapted to different temperatures. Represented with the blue diamonds are data from the Arctic sculpin, a fish that inhabits cold Antarctic waters like the ice fish do. Arctic sculpins typically live in waters around plus 2 degrees Celsius or so. The desert pupfish, represented by these red circles, is found in the desert southwest of North America. They frequently inhabit pools of hot water, typically at temperatures of 35 to 40 degrees Celsius or so. Okay, let's look at the data for hydrocarbon chain length first. The lengths of the hydrocarbon chains in brain phospholipids typically run from 16 carbons long up to 20 carbons long. We're plotting here the fraction of total phospholipids for each chain length. In nerve cell membranes from the Arctic sculpin, we see that the predominant phospholipid has hydrocarbon chains that are 20 carbons long. Phospholipids with shorter hydrocarbon chains are less abundant. In the nerve cell membranes of the desert pupfish, in contrast, the most abundant phospholipid has a hydrocarbon chain 18 carbons long, with 16 carbon and 20 carbon chains being somewhat less abundant. So this doesn't look too good for our prediction. We would have expected just the opposite pattern for the Arctic sculpin, namely phospholipids with the short-chain hydrocarbons should have been most abundant. In the more warm-adapted desert pupfish, 18-carbon phospholipids are more abundant than 16-carbon phospholipids are, but we would have expected a higher proportion of 20-carbon phospholipids, and we don't see that. Okay, what about saturation? Here, the data conform more to our prediction. We're looking here at three classes of saturation. Saturated, that is, with no double bonds whatsoever. Monounsaturated, in which there's one double bond in the hydrocarbon chain. And polyunsaturated, in which there are multiple double bonds in the hydrocarbon chain. In the membranes from the desert pupfish, it's the saturated phospholipids that are the most abundant, with fewer monounsaturated and polyunsaturated phospholipids. In the more cold-adapted Arctic sculpin, it's polyunsaturated phospholipids that are the most abundant. This is as we predicted. We can also look at the ratio of saturated to unsaturated phospholipids in a variety of different animals, ranging from the Arctic sculpin to goldfish acclimated at 5 degrees Celsius and 25 degrees Celsius, our desert pupfish at 34 degrees Celsius, and phospholipids from the rat brain, which typically live at the regulated body temperature for rats of about 37 degrees Celsius. Here, we see a quite regular increase in the ratio of saturated to unsaturated phospholipids as we go to higher and higher temperatures. Particularly interesting are the two goldfish ratios. When goldfish are acclimated to cold temperatures of 5 degrees Celsius, their ratio of saturated to unsaturated phospholipids is smaller than for goldfish, the same species, acclimated to the warmer temperature of 25 degrees Celsius. So this prediction, at least, is borne out by the data. Animals acclimated to warmer and warmer temperatures load up their cell membranes with ever more saturated phospholipids. In general, then, the hypothesis of homeoviscous adaptation of cell membranes seems mostly to be borne out. Animals adapt their cell membranes to the local temperature, seemingly to maintain fluidity around some particular value. This has relevance for how animals can adapt to cold temperatures, but it also has something interesting to say about the upper limits of body temperatures for animals. You'll recall that we've shown that there should be no upper thermal limit to enzyme function, but that nevertheless animals' body temperatures seem limited to the range of 37 to 45 degrees Celsius or so. This limit may exist because of a limitation of homeoviscous adaptation. As you go to higher and higher temperatures, as we've seen, membrane phospholipids become ever more saturated. But there's an upper limit to this strategy, because once a membrane phospholipid is saturated, it cannot become more so. It's impossible for the molecule to have fewer than 0 double bonds. It may be then that once nervous tissue has pushed homeoviscous adaptation to its maximum extent, further increases of temperature will increase membrane fluidity to the point where membrane voltages can begin to destabilize the membrane. This may be why the frank symptoms of so-called heat stroke and heat death are neurological in nature.